Hi, this is Ms. Bastido here to talk about Unit 4. We're finally on Unit 4. Unit 4 of QCE Biology covers genetics and evolution. So we'll be starting this in the first section of Topic 1, and this video will be over DNA structure and replication. So first we'll want to talk about what is the function of DNA. So you've probably heard of DNA, and you might know that you inherit your DNA from your parents. Who inherited their DNA from their parents. And so within that DNA, the function of DNA is it has a code in it, and we'll talk about what that code exactly is. But this code then determines the proteins that are made in your cell, and those proteins go on to give you various traits that you have. The other function of DNA is that it can copy itself and provide a copy of DNA to the next generation. And so this could be your individual cells dividing. So my skin cells are dividing pretty regularly. And so those cells are always making a copy of DNA before they divide so that each new cell has its own copy of the DNA. We'll get into more about the actual genetic code and how these proteins are made in another video. But we will get into the making a copy of itself in this video. So where is DNA located? So DNA is located, it depends on what kind of organism you are. So if you are a prokaryote, then DNA will be located in the cytoplasm. So prokaryotes, you might recall, don't have a nucleus or any membrane-bound organelles at all. And so their DNA is found in the cytoplasm. That DNA is naked and circular. And so we'll look at a picture of that in a minute, but because it's naked, it doesn't form chromosomes in the same way that we might find in eukaryotes. And also due to that, it loses a lot of functionality that we have in eukaryotes in terms of turning on and off genes, which we'll talk about in another video about gene expression. So in eukaryotes, we have DNA found in a few different places, depending on what type of organism you are. So all eukaryotes would have DNA in their nucleus, and in their nucleus would be found as chromosomes. And this DNA, we'll look at a picture of it, is going to be tightly wrapped around these proteins called histones to form chromosomes. Now, in humans, we have 46 chromosomes in every cell in our body, but every organism has a different number of chromosomes. And so you might have, for instance, a fruit fly has 8 chromosomes. And so they would have 8 chromosomes wrapped up to form around these histone proteins. And then lastly, we will have DNA in our cells that are not in the nucleus, but are in the mitochondria in humans or in any animal or plant, but also plants specifically also have DNA in their chloroplast. That DNA is also naked and circular and is really evidence for the endosymbiotic theory in evolution, which we'll get into later. But the DNA found in the chloroplast and the mitochondria is to make the enzymes that are used for cellular respiration and photosynthesis. And so it kind of skips the step of having to make that protein somewhere else and then transport it to the mitochondria and the chloroplast. It all happens right there in the mitochondria and the chloroplast. And so a picture of this is a bacterial cell, and this is this naked circular DNA. So when we say circular, it's not necessarily in a perfect circle, but if you were to stretch it out, it would be in a circle. So we say it's naked because it's not wrapped around those histone proteins, and it's not coiled up quite as tightly. And this can happen because bacterial DNA is not as big as the DNA found in, say, humans. So humans have 6 billion base pairs in their DNA, and so that's a big, big, big molecule. And so we have to wrap it up so it can fit into the cell. And then also in prokaryotes, you might find other little circular pieces of DNA that they've taken up over time, and these are called plasmids. So not its main DNA, but sort of like supplementary DNA that it gets from the environment. And so in eukaryotes, this is more what you'll see. So you see this is a chromosome, which you've probably seen a picture of. This is a 2-armed chromosome. Sometimes you'll see 1-armed chromosomes, but this one's a 2-armed chromosome. And this chromosome wraps up, so we've kind of zoomed out, or zoomed in here, sorry. And this DNA, which looks like a double helix, wraps around these histone proteins, and it actually wraps around 8 different histone proteins and then is secured by a 9th. Each of those is called a nucleosome, and those nucleosomes then continue to coil up around each other to form a chromosome. And so we call those supercoils. The purpose of the histones has a few different purposes, and the coiling has a few different purposes. It's a mechanism for turning on and off genes, which we'll talk about later, but also it's mostly that it can fit in the nucleus. The DNA would be, if it was all uncoiled at the same time, would not be able to fit into the nucleus. And then here you see here's a mitochondria, and they have these little circular DNAs right here in the middle of the mitochondria, in the matrix of the mitochondria. But you'd also find that in chloroplast as well. So this DNA in the mitochondria and in the chloroplast look really similar to DNA that you might find in a prokaryote. And so evidence that maybe the mitochondria, or evidence that the mitochondria and the chloroplast were once free-living organisms. And that's part of the endosymbiotic theory. So the structure of DNA was discovered by several people kind of working together. Actually, they weren't working together, but they were all working on a problem. And so Watson and Crick were working together. They were into model building and were looking at data, not necessarily their own, and trying to figure out what is the structure of DNA. So what does it look like? They knew that there were sugars, they knew there were phosphates, they knew that they were these nitrogen bases, but they weren't sure how it was all arranged and how that led to a genetic code. And so they were working on building models. I think one of their first models they built, Rosalind Franklin saw their work and thought it was, she knew it wasn't right based on the work she was doing. So she was doing X-ray diffractions, which help visualize the DNA in an X-ray. And Wilkins was also doing that, although they worked at the same university. They were not working together necessarily. But she wasn't really into model building, so she wasn't trying to build a model. She was trying to understand DNA better. And so kind of the short of this is that the Nobel Prize in 1953 went to Watson, Crick, and Wilkins for discovering the structure of DNA. Franklin was not given the credit for the Nobel Prize, but that is because she had already passed away from cancer, and so you can't receive the Nobel Prize posthumously. But her work was actually really important. So Watson and Crick saw a photograph without her permission that she had created of DNA, and figured out that the rungs of the DNA ladder, first of all, they discovered it was a double helix, not a triple helix, which they had previously thought. But then also they figured out that the DNA bases must be facing inward, and that inward helped them understand the rest of the structure. And so her work was used in order to come up with that model. And without her work, they would not have been able to come up with that model. That being said, the Nobel Prize was given to Watson, Crick, and Wilkins for discovering the structure of DNA. And so what is that structure? We can see here that the structure of DNA is that it's made up of repeating units, and these repeating units are called nucleotides. And so the nucleotide is made up of a phosphate, a sugar, and a nitrogen base. And so that nitrogen base can either be adenine, guanine, cytosine, or thymine. And we'll look at those in just a few minutes. The sugar is specifically, because it's DNA, is called deoxyribose. So when you see it drawn like this, one thing to note is every time you see, I think this is probably a better drawing, but every time you see kind of where it comes to a point, this pentagon, that's considered to be a carbon. And that's true up here too. So where it comes to a point, the carbon is implied unless there's something else written here. So this one has a nitrogen, so it's nitrogen, not carbon. But if nothing's written there, then we assume that it's carbon. And so that's just part of organic chemistry. And so obviously none of you have taken organic chemistry yet, so this is a bit confusing looking at these, but that's what that means. And so we can number these carbons actually 1, 2, 3, 4, and 5. That will become important later. So this is the first carbon, the second carbon, third carbon, fourth carbon, fifth carbon. We always start in the bottom right when we count those carbons. And so the basic structure is that there are these repeating units of nucleotides with a sugar, deoxyribose, a phosphate, and a nitrogen base, either adenine, thymine, guanine, or cytosine. You might have seen it before, A, G, C, or T. So the double helix, the way that it looks when it's, let's say if we were to unwind it, almost to make it look like a ladder, is that you have this sugar and a phosphate at the top, and then the rungs of the ladder are the adenines, thymines, guanines, or cytosines. And so the phosphate and the sugar are bonded together by a phosphodiester bond, which is a covalent bond. And so that's a pretty strong backbone, we would call it, of DNA. And then these other ones here are held together by hydrogen bonds. One thing you should notice here is that whenever there is a thymine, on the other strand, there's an adenine. Whenever there's a guanine, on the other strand, there's a cytosine. And so we'll look at that a little closer in just a minute. The other thing to notice by this picture is while this one is facing this way with the phosphate at the top, the fifth carbon at the top, 1, 2, 3, 4, 5, that fifth carbon's at the top pointing upward. Over here on this side, the carbon pointing upward is 1, 2, 3. And so this strand is upside down from this strand. And so we would say these strands are anti-parallel from each other. These anti-parallel strands are important when we talk about replication. And so it just refers to the carbons. Here you can see the carbons are actually numbered for you. But that fifth carbon, that's what we'd say. We call this the 5' end of DNA. And then the bottoms would be the 3' end because that's the third carbon sticking out. And then the 5' end on this side and the 3' end. So these are anti-parallel, meaning really one strand is upside down. And the way you can always just remember which side is the 5' end is look for the phosphate. So I always remember phosphate 5, and that should help you remember, oh, that's the 5' end. Although you could count the carbons as well, but it's an easy way to just sort of figure that out. So always remember that the DNA strands run in opposite directions, and so they are anti-parallel. So again, the bonds that are formed here, and so we would have a phosphodiester bond between the deoxyribose and the phosphate. And so in this one, we have several phosphodiester bonds. It would be between each of these phosphates and deoxyribose. And the same thing would be on this side. The bonds held in the middle are hydrogen bonds, and hydrogen bonds are relatively weak compared to these strong covalent phosphodiester bonds. So the hydrogen bonds are weak. That's actually going to be important because when we make a copy of DNA, we need to be able to unzip the molecule and break these hydrogen bonds. The base pairing in DNA, you've probably known for a while because you learned this probably in a younger grade, but adenine is always going to pair with thymine and cytosine is always going to pair with guanine. A few things to notice is that the reason that this is true is that adenine is a 2-ring structure, meaning it's a bit bigger of a molecule, but it's called a purine. So purines have 2 rings and pyrimidines only have a single ring. And so adenine always binds to thymine, but it's really a purine binding to a pyrimidine. Now, you don't necessarily need to know those words, but just being able to recognize kind of that one has 2 rings and one has 1 ring. And the hydrogen bonds between adenine and thymine are 2. They form 2 hydrogen bonds with each other, and the guanine and cytosine form 3 hydrogen bonds with each other. So a little bit stronger between cytosine and guanine as compared to adenine and thymine. So now that you know the structure of DNA, we need to figure out how does DNA make a copy of itself. And so it uses one strand as a template for the other strand. And we say that that means it's semi-conservative. And I'll go back into this later, but essentially you start with one DNA strand and you end up with 2 new strands, but each of those new strands are attached to one of the old strands. And so it always leaves a template behind because you always have the original strand that you started with plus the new strand attached. So the first step in replication would be to unwind and unzip the molecule. And so this is sort of a picture of it. You don't need to know about the binding protein, so you can kind of ignore that one. But this is the enzyme that you need to know about. So you only really need to know for this course 2 enzymes involved in DNA replication. There are many more, and I'll mention a couple of others, but you really only need to know 2. And so the 2 that you need to know are DNA helicase. Helicase unzips and unwinds the DNA by breaking the hydrogen bonds. That part is really important, not written here, but breaking the hydrogen bonds is actually how it's able to unzip the molecule. Keep in mind there's 6 billion base pairs in our DNA, and so we wouldn't just have one helicase molecule going from one end to the other. You'd have many helicase molecules moving across the DNA, and we call that a replication. It will form like a bubble of replication in that area. So it would be doing it simultaneously, and that will help allow for replication to occur more quickly. And so even here you see there's 2 different helicase molecules there. These enzymes unzip, unwind the DNA by breaking the hydrogen bonds, and that is the first step in replication. And so the second step in replication is for DNA polymerase, specifically 3, although you don't need to know that, it adds complementary base pairs. So if this is one, if I've unzipped the molecule and this is one side, you can see that I would have to be able to add the one nucleotide at a time. So this is the 5' end, and we build DNA 5 to 3. And so I would be building from the bottom to the top because the other side is going to be opposite. And so if I were to build that, we would say I would have, just trying to pull over this for you. All right, here we go. So we have a DNA polymerase enzyme. It's going to come in and start adding DNA nucleotides. And so what pairs with C? And so we know that guanine pairs with cytosine. And so it will bring in that nucleotide to base pair with it. And then as we move up, DNA polymerase will keep moving in the 5 to 3 direction. And then it's going to bring a nucleotide that base pairs with thymine. And so what base pairs with thymine? Adenine. And so adenine comes in and gets added. DNA polymerase keeps moving along the chain until it's formed a new strand, right? And so then we would end up, and it would keep going. I'm going to stop there. And so then we would have 2 new strands. Remember, they're antiparallel, and that we're building in the 5 to 3 direction. And so it's adding from the 5 to 3 on this side. Just remember the phosphate should always be on the side of where it's adding to. So it's adding from the 5 to 3 direction. And the reason we say it adds in the 5 to 3 direction is that it has to do with the enzyme's capability. So the enzyme is not capable of adding in the 3 to 5 direction. So it has to do with the structure of the actual enzyme and that how it has to work. And so that's our second step in DNA. The problem is with DNA, we have 2 strands. We had in the original strand, we have one that's 5 to 3 and we have one that's 3 to 5. And so because of that, on this side, it's quite easy. Just like I just showed you, we just read it and we build the other strand. But if DNA is unzipping in this direction, the problem with that on the other strand is if I can only build 5 to 3 and it's unzipping this way, I would be adding 3 to 5. And so what happens is it unzips a little way, and then DNA polymerase adds nucleotides the opposite direction. And then it unzips a little bit more, and then it adds a bit. Then it unzips a bit more, and then it adds a section. So we call these Okazaki fragments. You don't have to know that, but we do call these fragments. So on this, we call this the lagging strand. On the lagging strand, we'll end up with these little fragments. And on this side, it's just continuous, and so quite easy on that side. And here's sort of another picture of that. And you can see with the leading and the lagging strands that you can have these little fragments on one end. And so that's okay, but they do have to be connected eventually. And so there is another enzyme, you don't have to know it, but there is another enzyme that does that called ligase that connects the Okazaki fragments together. And so just as a reminder, DNA is semi-conservative. This allows for there always to be a template from the original strand. And so we start with 2 strands of DNA. We end up with 4 strands. Each of the new DNA molecule has 1 old and 1 new strand that are exact copies of each other. And the reason they can be exact copies is because we have this base pairing mechanism of adenine to thymine and guanine to cytosine. Hopefully that clarifies to you the function of DNA, the structure of DNA, and then how DNA replicates itself. If you have any questions, feel free to let me know.